that I did in the National Museum of Natural Sciences in the group of Borja Milan. And I'm going to talk basically about the speciation on islands, specifically about the genomic divergence between and within island population of the common chaffinch upon colonization of the Macaronesian region. So this just got accepted in the Journal of Evolutionary Biology, so I'm really excited about it, and I hope you enjoy it as well. So we were, why is it working? Okay. So we were interested in studying the genomic basis of evolution and speciation. So a good approach to study the speciation process is to use islands as model system. And why is that? Well, because insular populations are likely to suffer both drift and selection. Drift due to small founding populations, and selection because of the colonization of newer environments with new selective pressures. And therefore, islands are considered as natural laboratories for the study of evolution. And many of the best well-known radiations have occurred on archipelagos. For instance, the Darwin finches in the Galapagos, the Hawaiian honey creepers in Hawaii, and the Anolis lizards in the Greater Antilles. So depending on which evolutionary force is the main driver of the divergence process, either selection or drift, we can have adaptive or non-adaptive radiations. Adaptive radiations involve an ecological shift, and therefore selection is the main force driving this divergence process in order to adapt to the new or different environments. For instance, we have the Galula frogs in the Philippines that have diversified into 3 different ecotypes or habitat specialists in order to adapt to the different environments available. Non-adaptive radiations occur among clades with little ecological disparity, and therefore drift is the main force driving this divergence process. For instance, we have the Acatinella land snails in Hawaii that have diversified in nearly identical environments in the different valleys in the Hawaiian Islands due to non-adaptive factors such as drift or sexual selection. So we were particularly interested in understanding the role of natural selection in the divergence process, specifically in this interaction between selection and gene flow. So in the adaptive divergence process, when a species inhabits an heterogeneous landscape, it can spread to occupy all the different environments available. And the selective pressures in those environments will vary. Therefore, the divergence between these 2 populations will depend on the interaction between selection and gene flow. Moreover, the interaction between these 2 opposing evolutionary forces will depend on the magnitude of gene flow. That is determined by the dispersal capacity of the species and the geographic isolation due to barriers for dispersal. However, if selection is strong enough and dispersal is low or non-random, these 2 populations will eventually diverge phenotypically and genomically in order to increase their fitness at their own environment, resulting in local adaptation. So, in order to detect local adaptation, if we detect phenotypic differences at small spatial scale, we have to try to find the link between those differences, the genetic diversity, and the environmental heterogeneity. Several methods have been developed recently in order to study all these interactions. For instance, we have genome-environment association analysis between the environment and the genotype, genome-wide association studies and QTL mapping between the genotype and the phenotype, and common guardian and reciprocal transplant experiments to find the relationship between the environment and the phenotype. However, these last 2 experiments are difficult to perform, especially in non-model organisms. Another important factor to take into account is that most adaptive traits are polygenic and therefore leave this signature of selection spread along the genome because there are many loci of small effect. And this is usually challenging to detect. However, these methods, the genome environment association analysis and the genome-wide association studies, are powerful methods to detect this kind of polygenic selection. So I'm going to introduce our system, which is the common chaffinch in the Macaronesian region. So we generated genotyping by sequencing, which is a kind of a rat-seq. So we have multiple loci spread along the genome, and including the populations from the Iberian Peninsula, the north of Africa, and the three Atlantic archipelagos where the common chaffinch is present, including the Azores, Madeira, and the Canary Islands. So using this genomic data, we perform a phylogenomic tree using Raxanol, using over 100,000 SNPs, and we recovered that each population was a monophyletic clade. And we also observed that there was a sequential colonization from the continent, then the Azores, the Madeira, and finally the Canary Islands. This work is already published in Molecular Phylogenetic and Evolution. And we also observed that along this sequential colonization route, there was a gradual loss of effective population size and heterozygosity due to subsequent founder events, because each population was being founded by a population from a group of a small group of individuals from a population that was already genetically impoverished. So now, well, we think that this radiation was basically non-adaptive because due to drift among the different insular populations, because they all have a very similar environment. However, it's likely that in the first step of this colonization in the Azores, there was a strong selective process in order to adapt to this new insular environment, and those spread into the subsequent colonization steps. So now I'm going to focus on one of the last steps of this colonization, the island of La Palma. And the island of La Palma provides an interesting scenario, specifically to study local adaptation, because there are two contrasting habitats, the cloud and the pine forest. Both habitats are really different, both in resources and conditions. You see the vegetation is completely different, and the cloud forest is much more humid and dense, whereas the pine forest is much more open and dry. And here we wanted to test if the common chaffinch upon the niche expansion to the pine forest, because this is only found in this particular area, we wanted to test if upon this habitat shift, there were signatures of local adaptation that we could detect. So we sampled 5 populations per habitat. Here in the map, we can see the distribution of those populations. The light orange area corresponds with the pine forest and the blue area with the cloud forest. And we again generated genotyping by sequencing of those 10 populations. We also took morphological measurements. We also analyzed the feather-isotopic composition in carbon-13 and nitrogen-15, which is used as a diet proxy. And we also measure plumage coloration using an spectrophotometer. Besides that, we also analyze the environmental variables. Here you can see a map of the normalized difference vegetation index, or NDVI, which represents the vegetation greenness. And as you can see between both maps, there is a correspondence between the distribution of this environmental variable and the distribution of both habitats within the island. So now moving to the results regarding the differences in the diet using this feather isotopic composition, we detected differences in the carbon-13 isotope among habitats, being more depleted values in the cloud forest, which is consistent with more humid habitats. So these results confirm that they don't just inhabit different environments, but that they also eat different things. For instance, we have observed in the cloud forest, when we capture the birds, that they have paste, like berry paste in the bill, and in the pine forest, we have observed them eating directly from the pine cones. Then, regarding the phenotypic differentiation, we also found differences in morphological traits, specifically in the body mass and the wing length, and being larger and with longer wings, the individuals from the cloud forest. And this is consistent with the cloud forest being a more productive habitat. Then regarding plumage coloration, we also detected few differences in all the patches that we analyzed. And specifically in the breast, all the color variables that we analyzed were significantly different. Here you can see in these pictures that the individuals from the cloud forest show a darker orange plumage coloration in the breast. That is also consistent with Gloyer's rule that states that individuals from more humid habitats tend to be darker. Now moving into the genomic data. First, we analyze the neutral genomic structure. We perform a principal component analysis with almost 7,000 unlinked SNPs. And as you can see in this PCA, it corresponds with the geographic distribution within the island. So this suggests that the common chaffinch has a really extreme dispersal limitation within the island. His capacity to fly is perfect. So it's not that, it's just that they don't move around so much. And it was significant for isolation by distance, which means that the genetic differentiation is correlated with the geographic distance. So then we studied the habitat-driven adaptive divergence, and we implemented 2 genome-environment association analysis. Here I'm going to show you the results from the redundancy analysis that we used almost 10,000 loci and 2 environmental variables, the NDVI or vegetation greenness and the temperature seasonality. And here we got a really interesting result. Here you can see that the individuals from the cloud forest, that are the blue ones, separate from the pine forest individuals, that are the red ones, in the along the vegetation axis. And then you can also see that along this temperature seasonality axis, the pine forest population show a slight structure. So this pattern is interesting because first, we are able to detect this evidence of local adaptation at really small spatial scale and using a very small fraction of the genome. And also, this suggests that it might be something going on besides habitat adaptation to habitat at very small local scale. So we are trying to study more in detail this pattern. Then we also implemented bypass, which can be used either as a genome environment association analysis or as a genome-wide association study, depending if you use an environmental or a phenotypic variable. And then we perform an admixture analysis using the outlier loci recovered with this method, using the different variables, these 4 variables, the NDVI again, and then 3 phenotypic variables, the body mass, the wing length, and the bill width. And as you can see again, with the NDVI, we recover this structure, habitat-related structure. Then with body mass and body width, we find a similar pattern, but here 2 populations of the cloud forest show high probability of assignment to the pine forest cluster. And with the bill width, we find again a similar pattern of this habitat-related structure. However, now a pine forest population cluster with the cloud forest population. So if we combine all these outliers together, then we perform an admixture analysis, and we detected again this habitat-related structure, but this cloud forest population show higher probability of assignment with the pine forest population. So this again is suggesting that there might be something going on at very small spatial scale in the different localities, and we are trying to identify which are those variables and those factors that are causing this pattern. As we have the high quality reference genome and the annotation for the common chaffinch, we were able to identify the genes in those regions that show the different methods. And in these Venn diagrams, we can see the overlap among the different methods and variables. In the top one, we can see the overlap between both genome environment association analysis, the redundancy analysis, and the bypass using both environmental variables, the NDVI and the temperature seasonality. And as you can see, the higher overlap is among both methods using the same variable. And in the bottom one, we have the overlap between different methods using environmental variables and then phenotypic variables with bypass. And as you can see, there's also some overlap among methods and variables. So this helps us to identify the strongest candidates. And interestingly, among those candidates, we identified genes that were related with the phenotypic differences that we found among habitats. For instance, we have genes related with body size, with wing development, with plumage coloration, with parnofacial development. And we also identify genes that have been shown to be related with adaptation to high elevation and song learning. So now we want to try to find this if elevation or altitude has something to do with these local differences that we find at the specific populations. And also another PhD student in Borja Mila's group, Barbara Freitas, is now trying to identify if there is differences in song among both habitats. Because song and plumage coloration are both really important pre-mating barriers for birds, and this could promote and help the divergence among both habitats. So now we have generated whole genome sequences for 12 individuals per habitat. And we have already performed genomic scans with of different variables, including the FST or relative divergence, the XY or absolute genomic divergence, and genetic diversity. And as you can see in this plot, the FST peaks are coincident with drops in the absolute divergence and genetic diversity. And this pattern is consistent with the recurrent selection model, in which selection takes place in the common ancestor, and then those regions are subsequently selected in the daughter populations, leaving this pattern of reduced genetic diversity and reduced absolute genomic divergence, and generating these peaks in relative or FST, relative divergence or FST. And this lines up with the story of this sequential colonization of the common chaffinch, where probably the strongest episode of selection took place in the Azores. That was the first step, where they have to adapt to this environmental, new environmental habitat, that is the island. And then those adaptations spread throughout the following steps of the colonization to Madeira and the Canary Islands. We also did perform scans to detect selective sweeps. And here we detected an intriguing pattern that we are still looking into and try to identify what's going on here. So here are the selective sweeps. These peaks are the selective sweeps, and as you can see, the selective sweeps in the cloud forest population are coincident with drops in Tajimas D and high SNP density region, but in the pine forest population. And the selective sweeps identified in the pine forest population show exactly the same pattern, drops in Tajimas D and high SNP density regions in the cloud forest population. So at the beginning, we thought that this was a mistake. I repeated the analysis several times. I was really worried. And then we saw that it's like this. And we're trying to come up with an explanation for this pattern. And we hypothesized that it might be introgression going on, but we don't have the data to test this hypothesis yet, because we don't have whole genomes for the rest of the insular populations. We just have the GBS data, and with that, we cannot test it properly. So that's a following step to do, try to see if this is introgression from other islands. And that would be all. Like to conclude, like the common chaffinches colonize the Macaronesian region through successes founder events that cause this gradual reduction in genetic diversity and effective population size. We have seen that local adaptation through natural selection can overcome gene flow under conditions of reduced dispersal and strong selective pressures. So this process of local adaptation in the island of La Palma is particularly striking because the common chaffinch dispersal capacity is high and the geographical scale is very small. And as we see from this subsequent founder events, the effective population size and genetic diversity is really reduced. And islands are really a cool system to study the speciation process because they provide this window into evolution of small populations. And the loss of effective population size makes natural selection less efficient in island species compared to continental ones because they have this really genetic diversity of, that selection can act on. So in this paper in Current Biology, we showed this pattern, and in this graph we can see the insular populations that are the field dot have a lower effective population size and higher ratios of non-synonymous to synonymous polymorphisms compared to the continental ones. Here is included as well the common chaffinch. And this pattern of low effective population size and this high ratio of non-synonymous to synonymous polymorphisms affects both the ability of natural selection to efficiently remove weakly deleterious mutations and also reduces the adaptive potential of insular species. So this makes that this process of local adaptation and that we are able to detect this pattern using a reduced representation method of the genome really interesting. And hopefully these whole genomes that we generated would provide more insights in this process. And to finish, I would like to thank all the collaborators that were involved in this study, all the people that helped with the fieldwork, all the members of my lab, my tutor from the university, José Luis Bella, and both my supervisors during my Ph.D., Borja Milán and Rafael Sardoya, and of course the funding institutions for the project and my contract, predoctoral contract, and you for being here listening to me. Thanks.